Logan.
Do you know what my favorite thing about rats is?
What's your favorite thing about rats, Logan?
Well,
Rats are various medium-sized long-tailed rodents. All species of rats are found throughout the order Rodentia, but stereotypical rats are found in the genus Rattus. Other rat genera include Neotoma, Bandicoda, and Dioptimus. Rats are typically distinguished from mice by their size. Generally, when someone discovers a large muroid rodent, its common name includes the term rat, while if it is smaller, its name includes the term mouse. The common terms rat and mouse are not taxonomically specific, so rat is not a scientific term. Otherwise, all rats would belong to the genus Rattus. The best known rat species are the black rat and the brown rat. The group is generally known as the Old World rats or true rats, and originated in Asia. Rats are bigger than most Old World mice, which are their relatives, but seldom weigh over 500 grams in the wild. The term rat is also used in the names of other small mammals which are not true rats. Examples include the North American pack rats, a number of species loosely called kangaroo rats, and others. Rats such as the bandicoot rat are murine rodents related to true rats, but are not members of the genus Rattus. Male rats are called bucks. Unmated females are called does. Pregnant or parent females are called dams, and infants are called kittens or pups. A group of rats is referred to as a mischief. The common species are opportunistic survivors and often live with and near humans. Therefore, they are known as commensals. They may cause substantial food losses, especially in developing countries. However, the widely distributed and problematic commensal species of rats are a minority in this diverse genus. Many species of rats are island endemics, and some have become endangered due to habitat loss or competition with the brown, black, or Polynesian rat. While rodents, including rats, can carry many different zoonotic pathogens such as Leptospira, Toxoplasma gondii, and Campylobacter, the Black Death is traditionally related to have been caused by the microorganism Yersinia pestis carried by the tropical rat flea, which preyed on black rats living in European cities during the endemic outbreaks of the Middle Ages. These rats were used as transport hosts. Another zoonotic disease linked to the rat is the foot and mouth disease. The average lifespan of any given rat depends on which species is being discussed, but many only live about a year due to predation. The black and brown rats diverged from other Old World rats during the beginning of the Pleistocene in the forests of Asia. The characteristic long tail of most rodents is a feature that has been extensively studied in various rat species models, which subsequently suggest three primary functions of this structure: thermoregulation, minor proprioception, and a non-offensive meditated degloving response. Rodent tails, particularly in rat models, have been implicated with the thermoregulation function that follows from its anatomical construction. This particular tail morphology is evident across the family Muridae, in contrast to the bushier tails of the squirrel family Sciuridae. The tail is hairless and thin-skinned but highly vascularized, thus allowing for efficient countercurrent heat exchange with the environment. The high muscular and connective tissue densities of the tail, along with ample muscle attachment sites along its plentiful caudal vertebrae, facilitate specific proprioceptive senses to help orient the rodent in its three-dimensional environment. Lastly, murids have evolved a unique defense mechanism termed degloving, which allows for escape from predation through the loss of the outermost integument layer of the tail. However, this mechanism is associated with multiple pathologies that have been the subject of investigation. Multiple studies have explored the thermoregulatory capacity of rodent tails by subjecting test organisms to varying levels of physical activity and quantifying heat conduction via the animal's tails. One study demonstrating at a very significant disparity in heat dissipation from a rat's tail relative to its abdomen. In heat dissipation from a rat's tail relative to its abdomen. This observation was attributed to the higher proportion of vascularity in the tail as well as its higher surface area to volume ratio, which directly relates to heat's ability to dissipate via the skin. These findings were confirmed in a separate study analyzing the relationships of heat storage and mechanical efficiency in rodents that exercise in warm environments. In this study, the tail was a focal point in measuring heat accumulation and modulation. On the other hand, the tail's ability to function as a proprioceptive sensor modulator has also been investigated. As aforementioned, the tail demonstrates a high degree of muscularization and subsequent innervation that ostensibly collaborate in orienting the organism. Specifically, this is accomplished by coordinated flexion and extension of tail muscles to produce slight shifts in the organism's center of mass, orientation, etc., which ultimately assists it with achieving a state of proprioceptive balance in its environment. Further mechanobiological investigations of the constituent tendons in the tail of the rat have identified multiple factors that influence how the organism navigates its environment with its structure. A particular example is that the study in which the morphology of these tendons is explicated in detail. Namely, cell visibility tests of tendons of the rat's tail demonstrate a higher proportion of living fibroblasts that produce the collagen for these fibers. As in humans, these tendons contain a high density of Golgi tendon organs that help the animal assess stretching of muscle in situ and adjust accordingly by relaying the information to higher cortical areas associated with balance, proprioception, and movement. The characteristic tail of murids is also displays a unique defense mechanism known as degloving, in which the outer layer of the integument can be detached in order to facilitate the animal's escape from a predator. This evolutionary selective pressure has persisted despite a multitude of pathologies that can manifest upon shedding part of the tail and exposing more interior elements to the environment. Paramount among these are bacteria and viral infection, as the high density of vascular tissue within the tail becomes exposed upon avulsion or similar injury to the structure. The degloving response is a non-offensive response, meaning that it occurs when the animal is subjected to acute pain, such as when a predator snatches the organism by the tail. Specially bred rats have been kept as pets. Specially bred rats have been kept as pets at least since the 19th century. Pet rats are typically variants of the species brown rat, but black rats and giant pouched rats are also sometimes kept. Pet rats behave differently from their wild counterparts depending on how many generations have been kept as pets. Pet rats do not pose any more of a health risk than pets such as cats or dogs. Tamed rats are generally friendly and can be taught to perform selected behaviors. In 1895, Clark University in Worcester, Massachusetts, established a population of domestic albino brown rats to study the effects of diet and for other physiological studies. Over the years, rats have been used in many experimental studies which have added to our understanding of genetics, diseases, the effect of drugs, and other topics that have proved a great benefit to the health and well-being of humankind. The aortic arches of the rat are among the most commonly studied in murine models due to the marked anatomical homology to the human cardiovascular system. Both rats and human aortic arches exhibit subsequent branching of the brachiocephalic trunk, left common carotid artery, and left subclavian artery, as well as the geometrically similar non-planar curvature in the aortic branches. Aortic arches studied in rats exhibit abnormalities similar to those of humans, including altered pulmonary arteries and double or absent aortic arches. Despite existing anatomical analogy in the intra-athoracic position of the heart itself, The murine model of the heart and its subsequent structures remains a valuable tool for studies of human cardiovascular conditions. Laboratory rats have also proved valuable in psychological studies of learning and how other mental processes happen, as well as to understand group behavior and overcrowding. A 2007 study found rats to possess metacognition, a mental ability previously only documented in humans and some primates. Domestic rats differ from wild rats in many ways. They are calmer and less likely to bite, and they can tolerate greater crowding. They breed earlier and produce more offspring, and their brains, livers, kidneys, adrenal glands, and hearts are smaller. Brown rats are often used as model organisms for scientific research. Since the publication of the rat genome sequence and other advances such as the creation of rat SMP chip and the production of knockout rats, the laboratory rat has become a useful genetic tool, although not as popular as mice. When it comes to conducting tests related to intelligence, learning, and drug abuse, rats are a popular choice due to their high intelligence, ingenuity, aggressiveness, and adaptability. Their psychology in many ways seems to be similar to humans. Entirely new breeds or lines of brown rats, such as the Wistar rat, have been bred for use in laboratories. Much of the genome of Rattus norvegicus has been sequenced. Because of the evident displays of their ability to learn, rats were investigated early to see whether or not they exhibit general intelligence, as expressed by the definition of G factor and observed in larger, more complex animals. Early studies, circa 1930, found evidence both for and against such a G factor in rats. Quoting Galsworthy with regard to the affirmative 1935 Thorndike work, Robert Thorndike, for example, provided strong evidence for G in rats by use of a variety of tests such as mazes, problem-solving tasks, and simple avoidance conditioning. Performance tended to correlate across tasks with stronger associations found in mazes and problem-solving than with simple avoidance tasks. Thorndike also reviewed a dozen earlier studies which also suggested that the highest correlations are found between more complex problem-solving tasks. However, it should be noted that there were other contemporary studies that found split or near zero order correlation matrices for other populations of rats across cognitive boundaries. However, some more contemporary work has not supported the earlier affirmative view. Throughout the 1990s and into the 2000s, series of articles have appeared attempting to address the question of general intelligence in this species, though measurements of tasks performed by rats and mice, e.g., with statistical evaluation by factor analysis and seeking to correlate general intelligence and brain size, as is done in human and primates, where the general conclusion was in the affirmative. In 2011, controlled studies found that rats are actively pro-social. They demonstrate apparent altruistic behavior to other rats in experiments, including freeing them from cages when presented with readily available chocolate chips. Test subjects would first free the caged rat and then share the food. All female rats in this study displayed this behavior, while 30% of the males did not. Rat meat is a food, while taboo in some cultures is a dietary staple in others. Taboos include fears of disease or religious prohibition, but in many places the high number of rats has led to their incorporation into the local diets. In some cultures, rats are or have been limited as an acceptable form of food to a particular social or economic class. In the Mishmi culture of India, rats are essential to the traditional diet, as Mishmi women may eat no meat except fish, pork, wild birds, and rats. Conversely, the Musahar community in North India has commercialized rat farming as an exotic delicacy. In the traditional cultures of the Hawaiians and Polynesians, Rat was an everyday food for commoners. When feasting, the Polynesian people